Right, good evening everyone. My talk today will focus on some of the work that I've done during my PhD at the Department of Optometry and Vision Science. Our eyes are constantly exposed to UV radiation from the sun. Now in New Zealand especially, we're very much aware of how damaging these UV rays can be. They produce these particles known as reactive oxygen species that can interact with and damage every component of the cell. This condition is called oxidative stress. Now, so the tissues in our eyes are required to possess high levels of antioxidants like glutathione or GSH that can combat this oxidative stress and prevent any lasting tissue damage. However, as we get older, levels of antioxidants start to decrease, making the tissues of our eyes especially susceptible to oxidative stress and hence damage. So it's no surprise that eye diseases like cataracts and glaucoma are age-related. The tissues in the front of the eye, like the cornea, lens, and trabecular meshwork, are especially vulnerable. Now firstly, this is because of their positioning within the eye, because at the very front, they bear the full brunt of those damaging UV rays. In addition, these tissues don't have any blood vessels, so they cannot source their nutrients from the blood like most other tissues normally would. Instead, they rely on a body of fluid called the aqueous humor for their source of nutrients and, more importantly, antioxidants like GSH. Now, this fluid and all of its components are continuously being replenished by the ciliary body. Now, this, which ensures that there is a constant and fresh supply of nutrients for these tissues. The traditional school of thought is that the GSH in the aqueous humor is also produced solely by the ciliary body. However, interestingly, of all the tissues in the eye, the lens has the highest concentration of GSH. Now, of course, the lens requires some GSH to protect itself from oxidative damage. However, these high levels suggest that perhaps, like the ciliary body, the lens may also be involved in supplying GSH to the aqueous humor. So we hypothesized that perhaps the lens acts as a reservoir of GSH, releasing it into the aqueous humor so that the tissues in the front can then take it up. So my primary aims were firstly to investigate whether the rat lens was capable of releasing GSH into the aqueous, and this is called GSH efflux. So if this was the case, I then wanted to functionally characterize the mechanisms through which efflux was occurring. To address the first part of my objectives, I cultured pairs of rat lenses in artificial aqueous humor over different periods of time. Now this mimics the environment in which the lens would naturally exist within our own eyes. Then I collected samples of this aqueous humor to measure the amount of GSH in it. To do this, I added a fluorescent tag that binds specifically to the GSH, and then I measured the levels of fluorescence in each sample. So by doing this, I get a graph that looks like this one here. So if you look at the y-axis, that's GSH amount versus increasing time at the bottom there. So if I get you to focus your attention on the middle value or the median value of the data at each time point, you can see that the amount of GSH increases with time. So basically, this graph is telling us that the lens can and does release GSH into the aqueous humor. So having established that this efflux does occur, we then wanted to identify the mechanisms that could be mediating this efflux. Now, GSH transport across a biological membrane is mediated by membrane proteins called transporters. We screened for the presence, of 8 known GSH efflux transporters that specifically move GSH out of the cell. These have previously been identified in the kidney, brain, and lungs. So we wanted to do the same in the lens using a technique called PCR. Now PCR allows us to identify the presence of these transporters at the DNA level in the lens. So you can see that the presence of a transporter is seen as a presence of a black band. So of the 8 candidates here, only 4 are present in the lens. To further narrow down our choices, I then looked at whether these 4 transporters were present at the protein level in the lens, using a technique called Western blot. Now you can see that it's quite similar to PCR in that the presence of a transporter is seen as the presence of a black band. However, this involves looking at all the proteins rather than the DNA in the lens. So now out of the 4 transporters, only 2 are present in the lens, MRP1 and MRP5. The next step was to visually localize these 2 transporters within the lens using immunohistochemistry. Now what this basically means is that I take the rat lens, cut it up into very, very thin sections of tissue, and then add fluorescent green labels that specifically bind to the transporters, and then fluorescent red labels that highlight the cell membranes, like in this image. Now this is great because it allows me to orientate myself to the specific part of the lens that I want to look at. So what you see here is a capsule which covers the entire lens and basically holds it intact, then a single layer of epithelial cells that directly interfaces with the aqueous humor, and then the rest of the lens consists of several fiber cells. Now, for a transporter to be able to mediate GSH efflux out of the lens, I would expect it to be found at the epithelial cell membrane. Why? Because this is the cell layer that's the closest to the aqueous humor from the lens. Now you can see that with MRP1, it clearly and strongly labels the epithelial cell membrane. With MRP5, it's not as obvious. However, there does still appear to be some labeling in that image. So these images suggest that perhaps both MRPs could be mediating GSH efflux out of the lens. The next and final step was then to test whether those 2 transporters were actually functional. So I did this using an MRP transporter-specific inhibitor called MK571. Now, an inhibitor compound is one that specifically prevents the normal function of the transporter that it interacts with. So in this case, that's the MRPs. So once again, as you see, I cultured pairs of rat lenses in artificial aqueous humor, but this time in the presence and absence of the MRP inhibitor. Then I collected samples of the media, as I previously described, to measure the amount of GSH in it. Now, if the MRPs were actually functional, I'd expect to see less GSH efflux from the lenses cultured with the inhibitor compared to without the inhibitor. So I carried out 3 separate experiments, as you see here, and if we look at experiment 1 there, you can see that there is less GSH efflux from the lenses cultured with MK571 compared to without. The same pattern is seen for experiments 2 and 3, which confirms that GSH efflux from the lens is in fact mediated by the MRPs. So to summarize, I've shown that the lens is able to export GSH into the aqueous humor, and that this efflux is most likely due to MRP1 and or 5. Now once the GSH is in the aqueous humor, it can then be taken up by the tissues in the front of the eye. So you're probably wondering what the health significance is to all of this data. Well, persistent damage can occur to tissues in the front of the eye several years following cataract surgery and lens removal. This means that, traditionally cataract surgery has been performed on the elderly. However, with a growing diabetic population and hence an increased incidence of diabetic cataracts, more and more people are requiring cataract surgery at an earlier age. Now this means that there is a longer period of time after surgery in which damage to the tissues in the front of the eye can manifest. So my results have shown that the lens can supply GSH to the aqueous humor. Therefore, loss of this reservoir during surgery could reduce GSH availability to those tissues in the front, thereby increasing their susceptibility to oxidative stress. So it's important to consider the long-term effects of cataract surgery and perhaps look to finding therapies that enhance antioxidant levels in the front of the eye, thereby protecting against the accelerated onset of age-related eye diseases. Thank you.